Cellular respiration is a chemical process that generates most of the energy in a cell, supplying molecules needed to make the metabolic reactions run in an organism. There are two types of cellular respiration, anaerobic and aerobic respiration, which both undergo different processes to create energy in a cell. There are three stages of aerobic respiration: glycolysis, the Krebs or citric acid cycle, and the electron transport chain. There are two stages of anaerobic respiration: glycolysis and fermentation. The first step of both aerobic and anaerobic respiration is glycolysis because it does not require oxygen. Glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. Glycolysis takes the place in the cytosol of a cell. It starts with glucose and is then broken down by its own enzymes 10 times. One half of the reactions are part of an energy-requiring and energy-releasing phase. One example of this is the breakdown of fructose 1,6-biphosphate into 2-glycylaridehyde and 3-phosphate groups. Glycolysis results in the production of 2 pyruvates and 2 molecules, both of ATP and NADH. The Krebs or citric acid cycle comes next for the aerobic respiration and takes the pyruvate and breaks it down and creates compounds, resulting in the creation of carbon, ATP, FADH2, and NADH molecules. The last step of aerobic respiration is the electron transport chain. During this process, there is a power in the movement of hydrogen atoms into the intermembrane space. The number of hydrogen atoms, also called proton gradient, will build up and flow back to the matrix simultaneously, powering the production of ATP. The movement of molecules from high to low concentration requires no energy. This free source of momentum can be used as energy. In the case of the electron transport chain, the momentum is used to make ATP. 34 ATP are made from the products of one molecule of glucose. This electron transfer chain only occurs when oxygen is available. In total, in aerobic respiration, 38 ATPs are produced, 2 during glycolysis, 2 during the Krebs cycle, and 34 during the electron transport chain. The second type of cellular respiration is anaerobic respiration, which contains 2 stages: glycolysis and fermentation. Glycolysis had already been explained, thus showing the creation of pyruvates. There are 2 types of fermentation: alcohol and lactic acid fermentation. These 2 pyruvates from glycolysis form pyruvic acid. When sufficient oxygen is not present in the muscle cells for further oxidation of pyruvate and NADH produced in glycolysis, NAD+ is regenerated from NADH by reduction of pyruvate to lactate. Pyruvate is converted to lactate by the enzyme lactate dehydrogenase. Lactic acid fermentation converts the 3-carbon pyruvate to the 3-carbon lactic acid and regenerates NAD+ in the process, allowing glycolysis to continue to make ATP in low oxygen conditions. Since there is a limited supply of NAD+ available to the given cell, this electron acceptor must be regenerated to allow ATP production to continue. To achieve this, NADH donates its extra electrons to the pyruvate molecules, regenerating NAD+. Lactic acid is formed by the reduction of pyruvate. Alcohol and carbon dioxide are also created. Alcohol fermentation is the process in which yeast breaks down sugar into alcohol and carbon dioxide. Yeast are tiny single-celled fungi that contain special enzymes responsible for this reaction. Carbon dioxide gas bubbles out of the fermenting solution go into the air, leaving a mixture of ethanol and water. Anaerobic respiration results in the production of 2 ATPs due to the production of glycolysis where 4 are created but 2 are consumed, thus resulting in 2 ATPs. In cellular respiration, more energy is created during aerobic reactions. Thank you for watching. But just wait. I have a joke to tell. Of course. A mitochondrion walked into a bar and asked for a drink. The landlord served him the pint, and the mitochondrion asked how much it was. The landlord replied, "For you, 80p. You get it? 80p? ATP?